You are listening to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm your host, Peter Bannattini. Here, I interview neuroscientists and discuss their work, as well as the latest developments, issues, and controversies in the field of brain mapping. Today, I'm talking with Dr. Ahmad Hariri, whose work involves integrating psychology, neuroimaging, pharmacology, and molecular genetics in the search for biological pathways mediating individual differences in behavior and related risk for psychopathology. Dr. Hariri is a professor of psychology and neuroscience at Duke University, where he's also director of the Laboratory of NeuroGenetics. He recently was a senior author of an important paper that received attention both in the field and also in the popular media. And this paper, published in the Journal of Psychological Science in 2020 with first author Maxwell Elliott, is titled, "What is the test-retest reliability of common task functional MRI measures? New Empirical Evidence and Meta-Analysis." So we spend much of the podcast talking about this paper's implications and how we can address the challenges that it presents to continue to move the field forward. So in the podcast, I believe we have a lot of, I think, useful information, and ultimately the message is positive and hopeful. He's certainly not suggesting that we all switch our tools. fMRI is still extremely powerful technique and extremely useful. But he does talk about, we talk at length about, the sources of variability and the ways of actually moving the field forward in terms of, having more standard ways of analyzing the data, having standard ways of reporting the data, metrics for stability. And also the most important part, I think, is the idea that much of this lack of reproducibility comes from studies being designed specifically to look at group difference effects, as opposed to being designed to pull out individual difference effects. And so we talk more than a little bit about that throughout the podcast. So that's, I think, an important message to take home as well, that it's not only scanner instability or processing issues, it's how we design our paradigm. And that field is actually just beginning. So I hope you enjoy the podcast.
Okay, Ahmad, welcome. Welcome to the podcast.
Thank you. It's a pleasure to be here. Or be with you virtually anyway.
Yeah. One thing that maybe, one good side of COVID is that it made us realize that these meetings and this sort of thing is something, it's an option that we can do, and maybe continue to do a version of this,
Absolutely. Yeah. It's the new way of working, I suppose.
Yeah. So I knew you for a long time. I've known you since we overlapped a little bit at the NIH, and working with Danny Weinberger. And I always remember you going up to different things stand out. I remember obviously you had great work going on there, and we can talk about that. But I also remember at lunchtime, they had a gym at the top of Building 10, and you would go up and play basketball all the time there, and it was sort of like a regular thing that people did.
Yeah. Those were great times, Peter. I, like you, remember them very fondly, and met so many incredible friends and scientists. In many ways, those years were the most fun because I had very little responsibility in terms of supporting the research. That fell on Danny's very broad and capable shoulder. So I could spend my time downstairs in the basement doing cool studies. And then it gave me time to go upstairs and play basketball. And we played basketball with a lot of the ERDAs. Yeah. They really drove the games. But we got Danny Pine out there. We got Dennis Charney out there, and Terry Goldberg, and lots of the staff scientists and some of the really high-ranking program and branch directors came out and played. It was really fun.
That is so cool.
Those are good times. I don't know if I could manage a game these days, though, to be honest with you. But yeah, it was great. It was great. I really think that my career trajectory is a reflection of those years at the NIH. I learned a lot, and I was able to really push into directions that I had never even thought of. And that's a reflection of the general resources and research there, as well as Danny's specific program of research at the time. which really proved to be instrumental in my own development.
Yeah, no, I've always often tell people that doing a postdoc, I mean, it's great. Yeah, the NIH, all these opportunities, but still, right, doing a postdoc is sort of like academic nirvana, because you don't, like you said, you don't have to worry about anything. You just have to worry, and you're kind of at the stage you don't have to worry about getting your PhD anymore, and you should just do stuff.
Yeah, exactly. And when you have all of those resources at your fingertips, it's really a shame on you situation if you don't make the most of it. You have no one to blame but yourself if it doesn't work out in a way.
That's true. So let's just back up just a second. How did you, you started out, you got your PhD at UCLA with Susan Bookheimer, and you were at College Park beforehand at
That's right.
Park. But how did you start becoming interested in neuroimaging? I guess it was maybe at UCLA.
Yeah, it was actually slightly before I went to the West Coast to pursue my PhD in neuroscience. I was at Maryland at College Park, and as an undergrad, I was really interested in biology broadly and evolution in particular. And by the time my senior year came around, I really didn't have a plan. And so I scrambled last minute. This is, to confess that we don't all have everything planned out from the get-go. I scrambled and I was able to get into a master's program there in evolutionary biology. And so that was formative in a couple of ways. I worked in a wet lab where they were doing research with, one half of the lab was doing research with vampire bats and food sharing and vampire bats, and the other was looking at stalk-eyed flies and sexual dimorphism. And so what I learned from that was important was that I'm not cut out for wet lab work or field research. So that was good. But the really important moment for me was a graduate seminar on animal communication. And as part of that seminar, I learned about what at the time were these original initial studies on human language and it's the observation of human brain function in real time supporting language. And that just blew me away, Peter. I couldn't believe it. I mean, if I could choose to study anything, it would be human behavior. And here was an opportunity to look at the biological basis. The organ that creates behavior is now open to investigation. And that blew me away. And so, in fact, my connections with NIH go back before my postdoc years. Because I was at College Park just up the road, I reached out to a couple of labs to see if there was an opportunity for me to just come and help contribute to the research.
Yeah.
And Jordan Grafman was at NINDS at the time, and he said, sure. He said, sure, come on down. And he put me in touch with a couple of his postdocs and trainees, and I was able to at least begin to appreciate what imaging research was like firsthand. And I liked it. I really found it to be as powerful and elegant as the studies that I read suggested. And that's when I applied to PhD programs in neuroscience, ended up at UCLA. It was right at the beginning of the brain mapping center that John Mazziota and Art Toga created. And I was able to work with Susan Bookheimer, who remains one of my most important mentors, advisors, and friends.
Yeah.
And Susan was, as she is to this day, involved in a lot of different aspects of psychology and neuroscience, including imaging. And she really, it was funny. It's funny how so much of my career is serendipitous,
Yeah.
It's just just being open to opportunities that are presented. So when I got to UCLA, Susan had recently started a collaboration with Marianne Sigmund, who was an autism researcher. And as part of that research at that time, in the late 90s, face processing was really of interest. How is it that individuals with autism or on the spectrum process faces differently than typically developing people do? And that's what led to the development of this fMRI task, which, for better or worse, has been around for over 20 years and has been really widely adopted. And we can get to the worst part of it maybe later. But so that was how I really got to imaging. It wasn't certainly not like it would be today, where imaging is such a leading methodology in the study of human behavior. It was more on the fringes, and I came to it from an interest really in human behavior kind of broadly, rather than an interest in mental illness or any kind of clinical features.
Yeah. Okay. Yeah, no, that's actually, and you mentioned, right, you mentioned serendipity. I think that's a recurring theme. And so, I think, it seems like it's always serendipity. It seems like it's always sort of like, and then the people who are really good at recognizing the opportunities and making the most of them are the ones who sort of carry forward. And I think that, everyone has, tons of opportunity all around them. And it's just, right.
Yeah.
Sort of like making the most of that. But yeah, so at the NIH, I also remember you were definitely doing great work. And one paper that particularly caught my attention was the paper that's looking at basically it was papers published in Science in 2002 on the serotonin transporter genetic variation in the response of the human amygdala.
Right. Yeah.
It was really impressive. And it was sort of opened up, the possibility that you can see these individual differences and suggesting that you could, do that with imaging. So do you want to talk a little bit about that paper?
Sure. So I came to the NIH in part because I had grown up in that area. And I was ready to come back to the East Coast from the West Coast. And Susan Bookheimer had spent time at the NIH and gotten to know Danny Weinberger. And so when she learned that I wanted to go back to the area, she basically said, well, you have to meet with Danny Weinberger. Having no background or particular interest in schizophrenia research, I didn't know Danny or his work, but to this day, as I always have, I followed Susan's advice and I've never regretted it. So I came back for a holiday. I met with Danny, and within the first 10 minutes, I knew I would walk through, hot coals for this guy. He was amazing. Yeah. And the thing that really was important for me is Danny had this incredible program of research, many staff scientists, many researchers, postdocs, and he basically welcomed me into that program of research and allowed me to bring my interest in emotion and the amygdala and really fold it in seamlessly with his own research. And as part of that, it allowed me to begin to explore how genetic variation, common genetic variation, may shape the human brain and through that affect behavior. Yeah. And so that paper, which to this day is the most highly cited paper that I've ever published, it was a seminal finding, and it was a breakthrough for me in terms of my career trajectory because it allowed me to really carve out an important and kind of unique area, this integration of genetics and imaging. But again, it was really Danny's encouragement. He basically, I remember having a meeting with him and he said, hey, you've got enough data now where you've measured the amygdala using your task. Yeah. Why don't you go and see if there are any differences based on this genetic variant? Again, I didn't know this genetic variant from any other at the time. And sure enough, Danny's hunches were right. And what we found was that the version of this serotonin transporter polymorphism that had been previously associated with anxiety and depression was associated with higher amygdala response to these threat-related facial expressions. And so it at least suggested a mechanism. Yep. A biological mechanism through which a genetic variant may impact behavior by shaping differences in how the brain processes threat. We've learned a lot since then, right? One thing that's changed tremendously is that original paper, our original submission was with an N of 14, Peter, 7 in each of the genotype groups. Yeah. And Science, after the reviews, they said, hey, if you can replicate it, we'll be interested in publishing this. And so we had another 14 people, and we were 28 people, 28 subjects in total, Peter. Yeah. You could never do that work, and you should never do that work now. We know that single genetic variants are unlikely to have any kind of meaningful impact on brain function in the sense that it's going to be expressed downstream in behavior or clinical symptoms. Yeah. We've moved to the GWAS model. The Enigma Consortium has been really powerful in driving that forward, driving imaging and genetics forward in a more reliable and powerful way. And we, like most others, have shifted from any single variant model to polygenic scores. And there are pros and cons to that. One of the things that was really attractive about the single variants is to hone in on mechanism, right? So there's the transporter. We know what it does. We know where it's expressed. And if there's subtle differences in its expression based on a genetic variant, wow, that's telling us something specific about the biochemistry that shapes the function, the functional circuitry, and in turn behavior. The polygenic scores take us away, at least currently, from that kind of mechanistic viewpoint because they aggregate across the whole genome, across many variants that are unlikely to be directly functional.
Yeah.
To just generate but the signal is robust, the signal is reliable. So that's really the way that imaging genetics has gone. And I think for the betterment of the field and ultimately our understanding of how genes shape the brain and in turn behavior.
Yeah. And even stepping back a little bit, I mean, it occurred to me also that, using your evolutionary biology background, right. I mean, there's something like this that is selected for that might be useful evolutionarily to be more, aware and vigilant or anxious about certain things. But then at the same time, it has a flip side, potentially that affects, potential behavior in society. So it's interesting, sort of touches on things like that.
Absolutely. Yeah. And we could have a lot of fun talking about it, like this notion of group selection, that, the forces that shape the individual genetic variation may be actually open to group factors, right? So within any group, a tribe, let's say in our ancient history, it was probably beneficial to have some individuals who were highly anxious and highly vigilant for threat, because they could inform the rest of the group. Right. But you don't want everyone to be that way. You want some of them to. So David Goldman, who is, of course, a leading scientist at NIH, he had a model with another variant, COMT, where he called one variant the worrier gene and the other the warrior. So, again, these were all really attractive models and theories. The data really don't support the impact of any single variant outside of disease mutations. Of course, there's certain variants that are causal,
right?
But the ones that we are likely going to be able to leverage to better understand things like mental illness are not going to be of that ilk. They're going to be small effects distributed across the genome, but we just keep pushing forward. That's the best we can do, is let the science lead us forward. That's one of the things that I took away from my experience with Danny. He would always say, let the science trump. You may want this, someone else may want the other. Let the data take you forward.
Yeah, no, exactly. And I think that, right. And as far as the field is concerned, the data about replicability is something that it's interesting. A lot of researchers, including myself to some degree, have had a blind spot.
Yeah.
We always understood, it was a small effect. I mean, so let's sort of maybe transition into your more recent paper, but sure. and the idea is that we always understand there's variability. There's a small effect size. It's like a 1% signal change, and then if you try to modulate a task, then there's even less of a change you're looking for. You get good group results, and then everyone's trying to reduce the variability, and we can maybe talk about that maybe later in the context of the paper of manner, the subjects or responses,
absolutely
analysis, but everyone feels like, okay, well, it's close enough. We can still do something, and it's hard to get an intuitive feel of the statistics.
Yeah.
And your paper, I think, brought out, it kind of helped us hone that intuition.
Yeah.
Statistics. So, and so just the paper I'm talking about is your paper that came out in Psychological Science in 2020. What is the test-retest reliability of common task functional MRI measures, new empirical evidence and meta-analysis? And the first author was Maxwell Elliott. And so,
So I should certainly recognize Max's contributions here. Max is an absolutely brilliant graduate student. He's actually leaving for internship. He was an undergrad at Minnesota, and he worked in Angus McDonald's lab. But I think what was more relevant in terms of Max's contributions in this area is that Minnesota as a whole has long had a history, a tradition of measurement, of being interested in how we measure things. And this is something that remarkably captured Max's interest as an undergrad. And so he's a self-confessed measurement geek. And so he and my lead research analyst, Anshin Kanat, really spearheaded the work that is presented in this paper that was published last year in Psych Science. And just as a brief aside, one important way that we got to this step in our own research progress goes back to the serotonin transporter. One of the things that happened as a result of our paper is that a group led by Avshalom Caspi and Temi Moffitt published a subsequent paper in Science the following year showing one of the first demonstrations of a gene-environment interaction, which is very controversial in and of itself, right? Yeah. And so what they were able to show is that the same variant that we associated with higher amygdala response predicted greater depression in interaction with stressful life events. Okay. And again, everything just seemed to align so well, so powerfully. But the important thing was those two papers connected us together, and we met. We became very, very quickly good friends, and even 20 years ago started to fantasize about collaborating. They've been following a group of 1,000 people in New Zealand since they were born in 1972, and they've published incredible papers, just one after another across the lifespan. What early factors shape adulthood? What factors in adulthood shape later life? And now in midlife, what factors shape aging? Maybe we can talk about that at the end. But in 2015, I came to Duke. I was at Pittsburgh. They came to Duke. They kind of really lobbied me to join them, and I'm really glad that I did. But in 2015, we were finally able to get the funding to go down to New Zealand and collect the first set of MRI scans in this population representative birth cohort. Because it's a longitudinal study, we always had an eye on rescanning them, collecting multiple waves of scanning. And so that really put our feet to the fire when it came to the issue of reliability. Yeah. In other words, if we want to argue that changes are happening in these individuals based on physiology or experience, lifestyle, we need to be confident in our measurement of those changes. Yeah. And in fact, I'll share with you another kind of element of this. The first time we submitted the grant to try to get funding for this project, we went to a section that focused on epidemiology. And Peter, they tore us apart. And the main criticism was, and I don't know any of the details, but they argued, again, not without any specifics, that, well, it's becoming well known that fMRI is a wholly unreliable measurement. So why should we give you all this money to collect measures that we know are unreliable? And we thought, well, okay, let's see if we can understand this. And like you were saying, we have our blind spots. And the literature is so expansive that you can find one or two studies to support a particular, platform.
Right. Exactly.
Yeah. Which is really dangerous. It's a slippery slope. And we have to kind of guard against that kind of, kind of confirmation bias of sorts. Yeah. So, we were able to find, not surprisingly, enough studies that suggested decent reliability of task fMRI measures that we were able to get the funding. But one of the things we made certain of is, as part of this project, we wanted to collect test-retest data, and we did.
Yes.
And so it was really in the service of this longitudinal, long-term project of wanting to see how the brains of these people change over time and what that means for their health and their aging, that we got into test-retest reliability. I had no background, no knowledge of this. It's awful, it's shameful, but I didn't. I have to confess.
Yeah.
I've really learned a lot. And one of the important things that I learned in doing the work in our own data set, as well as through the Human Connectome Project data set and meta-analysis of the published literature, is this very old history described by Lee Cronbach of the experimental versus correlational model in psychology.
Okay.
And cognitive neuroscience, where I grew up, I cut my teeth, is based on this experimental model of psychology, where you want to minimize the differences between individuals in order to be able to maximize your opportunity to see a main effect, a group effect. Yes. And fMRI, task fMRI, has been incredibly powerful in illuminating how the human brain on average accomplishes any number of tasks, from simple perceptual tasks to more complex social tasks.
Yes.
It's great at that. And we have no qualms with that. And despite media reports otherwise, we have never intended to suggest that task fMRI isn't very well suited to do exactly that, to identify average patterns or reveal how the typical human brain accomplishes tasks.
Right.
But one of the things that I learned is that experimental model is built on design principles and assumptions that necessarily preclude or limit its applicability to looking at individual differences, which historically was part of this correlational model of research.
Yeah.
And as we kind of briefly, in the space that we had, suggest in our psych science paper, and we have unpacked it a bit more in a forthcoming paper in Trends in Cognitive Sciences. Just putting a little plug there.
Yeah, definitely.
Well, I think what happened for me, and I'll just speak for myself, is I saw these tasks producing robust group activations, and you run the task at NIH, you run it at UCLA, you run it in Berlin, you run it in Auckland, you get the same thing. You get these really robust group activation maps.
Yes.
And I made the assumption that robust group activation maps can be used to look at reliable individual differences.
Yeah. So just to clarify that a little bit, I mean, and with people, I think, and this is the part of your paper also that caused me pause, because I'm thinking, right, I mean, a lot of tasks are designed, like you said, to minimize individual differences so you can have a strong group effect. And it's interesting to start to think about, what type of tasks would stratify, would help to stratify differences.
Absolutely.
I don't think we've really begun, there's naturalistic movies, there's things like that, but it's not clear if there's a science of stratification.
They're certainly not in imaging.
Yeah.
And I would say not even in physiological measurements more broadly. Lauren Alloy, one of Lauren Alloy's PhD students, has published a paper where they introduced this term physiometrics. So in psychology, psychometrics is an entire discipline or subdiscipline. It's focused on exactly these questions.
Yeah.
Item response theory, classical test theory, generalizability theory. There are methods available, long-standing methods to try and design probes, design inventories, design measures that do isolate in a reliable way individual differences. It's just that it hasn't been a part of our work.
Yeah, no, I think in our work, our intuition is such that, you do something, and for instance, I'm just thinking in the analogy of running. Okay, you run a race, 5K or whatever, and then you'll have a certain distribution of times.
Yeah.
And sure, if you group, men versus women or something like that, you might get a group difference. And maybe the idea is that, well, with the 5K race, you get a certain distribution, but maybe you really pull it out with a 10K race or a 100-mile race or something like that, where things get more spread out, maybe something like that.
Yeah, that's a brilliant analogy, Peter, because one of the ways that the field is tackling this issue of reliability, if not directly, certainly indirectly, is through precision fMRI or pfMRI, where it's all about data aggregation. So much like your analogy, you probably learn more about the true running capacity of an individual the further you push them out. The more data, the more bold data we collect, the better the estimation of individual variation, individual differences in, let's say, intrinsic connectivity.
Yeah.
And we can talk about the pros and cons of this precision fMRI model later, but data aggregation is certainly one way forward.
Yeah.
And it's one of many that we and others are considering. I think with data aggregation, with precision fMRI, there's a very striking kind of a striking burden on participants, right? It's one thing to be in the Midnight Scan Club and to volunteer to be scanned for a couple of hours, or to be Russ Poldrack and, on a weekly or daily basis, scan yourself. It's another thing to get, to take individuals with autism or individuals with schizophrenia or even kids and get that kind of data. There are very important cases where precision fMRI should be used even now, like in localizing targets for DBS or other forms of neuromodulation.
Yes.
But one of the things that are dovetailing with reproducibility, replicability, reliability is to move imaging into population representative studies where we can really understand the true effect sizes that can be expected. And I don't think we certainly can't go back down to New Zealand and scan our 900 study members for 2-3 hours. It just, we couldn't afford it. They wouldn't do it. So while data aggregation is one approach, I think there are others that may be even more applicable and useful in the larger context of studying, the human brain.
Yeah. So I got ahead of myself a little bit. So definitely,
it's okay.
So I definitely will. That is a great discussion of what the solutions are. But let's just back up just a little bit. So the main finding of your paper, essentially, the main thrust to your paper, you use a term that everyone is learning, if they don't know already, is the interclass correlation
coefficient. Right. Yep.
It's basically, without going into too much detail about it, it's basically inversely proportional to like the group distribution in some sense.
Yeah.
And there's different ways of measuring it, or different types of data you can use to measure it. But with fMRI, you really lay this out nicely in this paper. And you say,
thank you, Peter.
Oh, sure. Well, no, it was a really nice paper in that regard. And it looks like your paper was two parts. You scoured the literature, which seemed like it was just a ton of work. And then you looked at these large group data sets as well. But, the findings were a little bit more than a little bit disturbing in some sense, where the ICC necessary for like making statements with the number of subjects is, it has to be pretty high. And you found that most studies had about an ICC of 0.3 or something like that.
Yes, sure. Yeah. We were disturbed more than any other. And in fact, I remember early on having conversations, if not with, my immediate colleagues, in my own head about like, what do we do with this? We know this now. At minimum, we knew that we were not going to continue to publish individual differences analyses using task fMRI. And in fact, if you look at our publications for the last 3 or 4 years, other than papers that were in the channel, we focused on structural measures because of their relatively high reliability.
And that's interesting in itself. Like why is it, that jumped out at me in the paper that somehow the variation in structure is much less, but it is more telling. It does vary though with disease.
Yeah, we uniformly find ICCs, these test-retest reliability. In other words, how consistently does a measure reflect the underlying target or construct, the true score versus the error score? In structures 0.9 or higher, cortical thickness, surface area, subcortical volumes, even, FA. It's amazing. It really is. And you would know much better than I would, Peter, to what extent that reflects the precision with which, we can measure structure versus function. Clearly, there's more noise when we're looking at the bold signal.
Yeah.
But for that reason, I think we currently lean heavily on structural measures in our work. We can talk about this later. We've transitioned to becoming interested in midlife brain structure and what it can tell us about an individual's likely risk for developing Alzheimer's and other dementias as they age.
Yeah.
And in that literature, the more clinically kind of applicable measures are all structural.
Yeah.
Cortical thickness, hippocampal volume, white matter hyperintensities. These are all structural features of the brain that have, at least in comparison to task fMRI or even resting or intrinsic measures, have reached a level of at least being clinically informative, if not diagnostic.
Yeah.
And I think that can't not be related to their higher reliability.
Yeah, exactly. So going into that sort of is a good lead into all the sources.
Right.
Like why is it that fMRI is somehow is less reliable? And it's interesting. It's near and dear to my heart because I in the context where I'm a core facility director, we have a researcher who has 250 subjects or something like that, and it's a very subtle task of looking at modulation of it's like a gambling task, and it's looking at depression, and we're having a hard time. It's like he came to me and said, "Oh, look, our ICCs are really low," and the initial thought was there's a problem with the scanner. And we check the scanner's fine. And could it be could it be shimming? Could it be like the placement of the ROIs, like that variability? And I think we're starting to arrive at the thought that, certainly, you have different subjects, you have all kinds of variability in their physiology.
Sure.
And also with the pre-processing too. There's all kinds of sources of variability in that. We started to get to the idea that, it seems like the subject response variability might be driving this. So it's not an MRI thing necessarily. Certainly these variables are bad, but it seems like during the study, subjects don't always behave as you expect them to. And it seems like monitoring that was potentially explanatory of this. I don't know what you think of that.
I certainly think that accounting for that kind of variability it will help improve the reliability of estimating brain activation.
Yeah.
But honestly, and this is a question that came up when our paper was first published, especially from different media outlets, is it a problem with the scanner? And I would absolutely say no. This is not a problem with MRI. It's a problem with how we are applying MRI. And I think it really falls much more on the side of the tasks and how they were developed. Again, going back to this experimental versus correlational framework.
Yes.
The vast majority of tasks that I'm familiar with, whether it's our FACES task or the monetary incentive delay task, they just were never developed from the ground up to look at individual differences. This is not only task fMRI, by the way. Like the Stroop task has relatively poor test-retest reliability at the level of individual. It's right around 0.4 as well.
Yeah.
And this is a task that's been used in psychology for decades, right?
Yes.
But not developed to look at individual differences, but at how the average human brain deals with incongruity and resolves incongruity.
Yes.
And what can we do? I think we can do a lot of things, and certainly working with you, Peter, and other experts in the technology is going to be important. One thing that we're excited about, at least in terms of the potential, is multi-echo fMRI.
Okay, great.
So Chuck, you may know, I mean, you definitely know this better than I do, but Lynch and colleagues, Chuck Lynch and John Power, working with Faith Gunning and Connor Liston at Cornell, published a really incredible paper in Cell Reports where they showed that 10 minutes of multi-echo produces much better, much more reliable measures of intrinsic connectivity and even task-based patterns of connectivity than 30 minutes of single echo data.
Yes. Yeah.
And so, that gives us hope that we can perhaps accomplish precision measurement in far less than, 90 minutes, 120 minutes, 3 hours.
Yeah.
And I think multi-echo, again, I'm speaking out of school here with you, Peter.
Well,
I think part of it is minimizing some of the noise, the non-physiological bold noise. And if I understand correctly, you can even tweak it to make sure that you're targeting certain regions of interest, like the amygdala or the thalamus or the cerebellum, in a way that you get better signal.
Yeah, you can certainly use the shorter echoes in the areas that have susceptibility dropout.
Yeah.
Yeah, no, actually, well, obviously, I'm definitely a huge proponent of multi-echo.
Yeah, and that's something, if we're going to, well, I shouldn't say if, but when we continue our fMRI research in terms of new data collection, we're absolutely going to use multi-echo. Unless something even better is available, it's clear to us that multi-echo is the way to go.
Yeah.
And so what I hope we've been able to do with our paper, Peter, is just to bring a higher level of awareness to this limitation. Some people don't really want to acknowledge it. They want to just keep going forward, business as usual. We absolutely are not going to do that. And I don't think we're going to get anywhere if we continue to do that. Now, what we hope is, having brought this to a higher level of awareness, many people like yourself and others are going to pitch in and help figure out solutions. I don't think it's going to come from one source. I think it has to be from the technology side. It has to be from the stimulus design side, from the observation and measurement of individuals over time, understanding, intrinsic sources of variation and accounting for those, modeling approaches. It's a big problem, so it's going to require a lot of pain to solve.
And I think it's important to have that point come across, that your paper isn't saying stop doing fMRI. It's just saying, I think a lot of people interpret it as like, oh, well, people worry that, our grants aren't going to get funded if we even mention fMRI, because now
we're worried about that too, by the way. This is not just, it really is our worry as well. Yeah.
But I think it's an important point, to say, look, maybe multi-echo, maybe better reporting metrics of time series stability. Absolutely. So you can actually then go back and sort of maybe bin, not really in a biased way, but sort of in an objective way based on the stability. Understanding, even some people even argue that, and this is sort of along the lines of stratification, some people argue that, like in the ABCD study, you have these pre-selected categories of subjects. Right. Maybe if you let the data speak for itself, the categories may not be the correct categories, and you might have data-driven categorization that will pull out an increased effect size.
That gets us right into the heart of psychometrics, which is allowing the data to tell you something important about the measure.
Yes.
So item level response theory, this can be applied to imaging data.
Yes.
You can do split half reliability of a scan. You could do item level modeling.
Yeah.
Late variables, I think, are going to be really powerful in helping advance reliability.
Yeah, and also, right along those lines too, I think that's one thing that your paper mentioned, but it didn't. It's really hard, obviously, to do the analysis of. But I think multivariate analysis of the fMRI data has a chance to sort of leverage a little bit more power in some regard.
And there are papers out there that show better reliability with multivariate analyses or, multivoxel pattern analysis. There are a number of ways to go about it. And I think those are great. It's one way to make the most of the imaging data that so many of us have already collected, and to move that forward. I think that's true. The other thing that we have done and others are doing is we're basically combining all the bold data in any given data set, be it task or resting state, just to serve the function of data aggregation.
Yeah.
Max Elliott published a paper in 2018 showing that if you combine task and rest data, both in the Dunedin study and in the HCP data set, and then you estimate intrinsic connectivity, you get more reliable estimates with the concatenated kind of total data set, which he calls general functional connectivity, than you do with certainly the task alone, but even with rest alone.
Yes.
We understand that the need to be able to use the data we have on hand in the most effective way. Beyond that, I think we have to push to new approaches, adopt new strategies. That's going to require an investment on the part of funders because we don't know. We don't have the answers. We have ideas. There's suggestive evidence available now, but we need to dedicate a lot of effort and resources to better understanding reliability and developing strategies where we can maximize individual differences. If we have any hopes of clinical translation, I think this has very little to do with cognitive neuroscience in the traditional sense. I think task fMRI and small n studies can still help us drive our understanding of the typical human brain, of what the human brain is capable of and how it's capable of creating behavior and lots of other things.
Yeah.
But we really have to pivot. We pivoted very kind of casually and in retrospect, carelessly from that model to the individual differences model.
Right.
And now we really have to acknowledge that kind of mistake in a way, or oversight, and we have to move ahead in a way that's much more systematic.
Yeah. So it also just occurred to me just now that I'm trying to think back of some studies that seemed like they strike me as more convincing in some studies. And it seems that there have been a lot of studies that, show intelligence, like, IQ. And they seem like they show good results. Well, maybe that's because these IQ tests have been designed to pull apart individual differences better.
Absolutely. In fact, in psychology, the gold standard for test-retest reliability of measures is IQ. And it's because there have been decades, if not centuries, of investment in IQ testing and especially at specific items that will tease apart differences between individuals in their intelligence.
Yes.
That is exactly what we need to bring to bear when we want to look at brain, when we want to look at brain function in particular.
Yeah.
Yeah. IQ is the perfect example. I for one, and I know others are very, very confident, or at least very optimistic in the applicability of those types of measurement principles to imaging.
Yes.
I know it's happening. I see in the literature, like the Lynch paper, that takes a slightly different approach, but we've mentioned already the naturalistic stimuli, movie viewing. There's data that suggests that type of experience in the scanner is not only more ecologically valid, is closer to our real world experiences, but it actually helps us identify more reliable individual differences.
Yes. Yes.
It doesn't constrain individuals to process information in a very rigid experimental way.
Yeah, and also movie viewing is more efficient. I mean, it's sort of packed with this information, and it's tricky to analyze. I mean, you can do, there's different views of how to properly analyze that, starting out with models of various aspects of the movie, or just looking at cross-subject correlation like Emily Finn.
Yes, that's a great example. Great example. But it's interesting that there are already now a number of fully annotated movie stimuli that can be adopted. In addition to doing multi-echo, we are very strongly, considering doing naturalistic stimuli, because our goal is individual differences, is to measure individual differences. So we are going to bring as many of the available strategies to bear on this issue, and hopefully create a new model, a new paradigm that is exportable, that can be adopted and lead to translational findings.
But you have like a metrics, so you have the ICC, which is a great metric.
Yeah, there's internal consistency, right? So there's Cronbach's alpha, that's good. And honestly, the more measures of validity and reliability, the better. One of the things that we suggested in our psych science paper is that we should adopt as a field a standard of reporting these measures in any study that's focused on individual differences. So if you want to show that activation, be it multivariate or be it a task ROI, a main effect contrast value, if you want to show that that is relevant for understanding, let's say, anxiety or an individual's future risk for depression, then I think we have to have minimum reporting standards. Yes. So the reader can interpret and be able to conclude, okay, how valid are these conclusions, right? That I think is a minimum that we really need to expect. And I've heard from various journals and editors that they are seriously considering adopting these standards like they have for replicability, right? So their journals now, if you want to publish a genetic association study, you better have at least one replication sample.
Yes.
It really needs to be an independent replication sample. I think again, pushing imaging and pushing functional imaging to translational and clinical applications and biomarker development, these are minimum standards we have to adopt.
Yeah, I completely agree. But do you think, and we talk a little bit about the clinical application of fMRI, certainly, and the reason why pre-surgical mapping works is just it's just like a main effect, and so you just see something. It's you're not trying to tease apart differences.
That's right.
And also, as you mentioned, sort of maybe looking for places to neuromodulate in that regard, or even epileptic foci or things like that might be useful. Yes. But as far as like using, let's say we even have a pretty good measures of individual differences.
Right.
With a large enough data set. Yeah. And let's say, so to actually translate that, like coming up with it would be, like one of the holy grails of fMRI, right, is to have templates. Right. That's sort of like this is the activation pattern that you need to see to predict this treatment with a drug, or, to help differentially diagnose this person with anxiety. And you differentiate in the paper between individual differences and clinical applications.
Yeah.
Do you think there's hope for that? I mean, even if we can.
Absolutely.
Yeah.
Absolutely. I do. I as much as it may appear from that paper last year that we're giving up on task fMRI, that couldn't be further from the truth, Peter.
Yeah.
We want to do it better. We need to do it better. And so we have accepted this challenge.
Yeah.
I think we can get there. Now, it's interesting. Where these kind of clinical cutoffs come from is a bit of a mystery. There are like literally a handful of papers that have established these kind of criteria for what's poor, what's average, what's good, what's excellent reliability, and what should be clinically applicable versus not. So like 0.8 is often used as an ICC of 0.8 is like, okay, this is a clinically meaningful measure. Now, generally speaking, of course, we want the reliability to be as high as possible when we're making decisions about an individual's health, right? So we need to know that measuring ambulatory blood pressure, we can do it reliably because we're going to decide whether we give this person a hypertensive medication based on the results. Yeah. Now, we are moving forward with kind of, again, based on, and unfortunately kind of more of a historical legacy of like a 0.75 or above, even 0.6 or above, I think is at least workable. As long as we're reporting what the reliabilities are, I think transparency is key. Yes. And then it'll allow the field to move forward more confidently. Yeah. But I really think that studying reliability in and of itself is something that we need to become better at, right? So in the Dunedin study, we looked at test-retest in 20 participants. In HCP, I think it's 45. Is that enough? Yeah. Probably not. Should we do it in larger groups, more variable groups of individuals? Absolutely, right? So I think that just kind of the measurement of reliability and what the parameters really should be, how we optimize the parameters in terms of sample size, the interval, what's the right interval? Is it a week? Is it 2 days? Is it a month? Yeah. We don't have the answers. And those are really basic questions that I think, again, if we don't answer now, we might regret 10 years from now. Yeah. But we say, oh, look, well, all our test-retest work was based on an interval of 2 months. And now we know that 2 months is too short or 2 months is way too long. Yeah, it's a challenge. Like I said, it's a big problem. But I for one am committed to helping solve it. I have no confidence that we individually will solve it, but I have great confidence that collectively, we can do it.
Yeah.
We've done, great things as a field, and I'm confident we'll continue to do it with task fMRI.
Yeah, and I think that your paper really did sort of tip the field to say, hey, wait a second. And obviously there's many other papers that have been sort of pushing in that direction, but I think your paper was nice in the sense it sort of laid it out. And I think that we're just at the very beginning of really understanding the nature of the problem. It's clear that there probably will be a solution as far as this is concerned. So it's got to do the work.
It may be closer than I even realized. Like, again, the multi-echo stuff is phenomenal.
Well, I'm glad you say that.
Your group has been involved in that development, right, Peter?
Yeah, no, we've been pushing it. My graduate student, Prantik Kundu, developed the whole multi-echo ICA. So the issue with multi-echo is that, we're still trying to figure out the best way to analyze multi-echo. So, but he pushed these really nice ways of looking at multi-echo. And definitely, I totally agree. I'm completely in the multi. We've been developing I've been doing multi-echo for 30 years, since the beginning almost.
So Peter, I'm curious, like, if we could turn the tables a second, why hasn't it been adopted more routinely and more rapidly?
Yeah.
Hardware question.
So this is another one of my sort of things that I sort of rant on sometimes, but and it's the whole idea that we use clinical scanners for fMRI, and it's been hugely beneficial to the field to do that. However, clinical vendors, they're focused on the clinical market. And fMRI is not a clinical market, and so they don't really make it easy for people to develop and distribute these novel pulse sequences.
Gotcha. So they don't come stock with when you buy the Siemens Prisma or the latest GE scanner, they don't come as part of the stock sequences.
No, not yet. And unless there's a different clinical application that's already there for multi-echo, they might develop it and they might allow people with research agreements to share it, but it really hasn't permeated the field enough. And I totally agree with you that if there were only a mechanism by which the vendors were a little bit more open to sort of having more of a platform for catalyzing these clinical applications of fMRI, that's probably one of the big reasons why fMRI hasn't sort of entered into the clinic, even for the other things as much, because it's just, you have to have these custom-made environments for doing fMRI, which most clinicians just, you want to push a button and see the results, and it's hard.
Well, I mean, if what is happening now will help with that push, to have the big clinical vendors adopt multi-echo, because it's like this old adage is like, we'd hire you, but you don't have enough experience, right?
Right. Well, how do you get experience?
We'll load multi-echo if it has clinical applications. Well, we need to have multi-echo to demonstrate clinical applications.
And this is the nature of a lot of my discussions with vendors for the last 20 years is exactly this. And to their credit, all the vendors have a version of multi-echo, I think, right now. But it's just not, we have this website called TDANA, which is TE data analysis. And people can use these packages for processing it.
Oh, that's great.
And so I think that hopefully with people like yourself mentioning the results of this, it would be great. Because I think actually, I've had good discussions with other groups who, like the Connectome data set, their track is not to do multi-echo, but was more to do very short TR. Okay. And their thought was that if short TR, you can model the noise better and get rid of it better. So, and that was an argument for a while, and I think that's true. But I think now we can have both. With there's fast imaging sequences, we can have short TR and multi-echo to some degree. Yeah, I really do think I agree with you that multi-echo is a hugely powerful thing because it gets directly at susceptibility contrast fluctuations.
Fundamentally, what we need to do is better isolate true score from error, whether it's from the acquisition side or the stimulus side. That should be the ultimate goal, and we're getting there. Again, I am very much encouraged by what's already happening in the last couple of years, and so hopefully we can, when we get back down to New Zealand and start to scan study members, we can get you on board to help us make sure we're doing multi-echo the best possible way, Peter.
Sure. Happy to help. Definitely. Yeah. So, okay. So that's good that there's definitely hope on this.
Absolutely. It's, I'm not about doom and gloom. I'm an optimistic person. And I think we just push forward. We just keep working hard. The thing we can't do is ignore it.
Yeah.
We can't bury our heads in the sand and say, you know what, I've got findings. Like, in 2015, we published a paper in Neuron where we showed that the amygdala response in a fairly large group of young adults, several hundred young adults, variability in that signal was predictive of their later reporting of depression and anxiety. And we thought, wow, maybe we've got a biomarker here.
Yeah.
But of course, now we know that we got lucky, basically. I mean, there's no way of really getting around it. We captured this in our data set, but now knowing the reliability of our task, we have no confidence that it can be exported to do so again and again in a reliable way in different populations at different times in order to have prognostic value. And that's ultimately what we want. Is we want to be able to predict how individuals will evolve over time and how their experiences will shape that evolution through the interaction with their brain.
Yeah, yeah.
But so, part of the issue that we certainly don't have time to get into here is, why are there so many papers showing the amygdala is associated with anxiety or variability in stress responsiveness? There are a lot of papers. There's a file drawer problem. I think we know about it if we don't address it directly. I think there are a lot of null findings that just never make it into the literature. And another kind of historical legacy of task fMRI is I think there has been a way too permissive environment for analysis.
Yes.
I'm going to take the max voxel. I'm going to use the mean cluster. I'm going to do a 5-millimeter sphere around that. And you know what? We've been getting away with that because we don't have a standard. We need a standard.
Exactly right. Yeah.
It's very difficult to see that kind of continued strategy ever resulting in clinically applicable measures, because you're not going to get the same max voxel in your data set as I get in my data set. So it's all
I totally agree. And actually, that's what OHBM, I just had an interview with people at the standards committee, trying to come up with best practices that we can all agree on, and not without making it rigid and prescriptive, but at least having something that we can base.
Absolutely. Yeah. And certainly if we can get OHBM, of course, to promote it. And then I think we need to get the best journals and the editors to appreciate the importance of it and to really expect it from papers that are submitted for publication.
Yes.
I think that ultimately is going to determine whether people adopt it or not adopt it, whether we can establish standards. It's time, right? It's overdue to have these standards. And I'm not saying this because I'm trying to sling mud at others. We've done this.
Right.
Our analytic framework has changed
with
time. And structural measures have have been so much more solid in that sense, right?
Right. There's less parameters, though. There's
less parameters. Absolutely. It's easier to do, for sure. But, I think it's wonderful that OHBM is pushing for this because I don't think we're going to make the kind of progress that we all want and hope for without those standards.
Yeah. Well, that's great. I totally agree with that. And we can't do this fast enough, but you're right. It is a sort of balance because, we're all trying to figure out what the best thing to do is. We don't want to cut off possible avenues, but at the same time, we have to have some way to have an objective way of assessing.
Yeah. Yes, absolutely, Peter.
Well, cool. This has been a really important, and I really think that this is a great discussion on everything from your results to just one thing to sort of end this one small part with as we ramped out is that there's still people who do small N studies, and there's still a purpose for. There's still a place for that.
Sure. Absolutely. For me, those small N studies, including many that I conducted early on in my career, gave us the first inklings of where to look, what's happening. Again, if the listener leaves our conversation with anything, I hope it's that task fMRI has been such an incredibly valuable tool. And we've learned so much about the human brain. We are not in any way trying to impugn the importance and value of that. What we want to do is we want to be able to transition, to leverage, to pivot from that in a more careful and kind of optimized way to be able to move into individual differences in those very basic human processes that fMRI has been absolutely seminal in revealing.
Yeah. Well, I'm excited to see how things will keep on going forward.
Me too, Peter.
So is there anything you want to part with? It's been fun to watch your career progress. You're extremely, you're doing amazingly well and you're,
I'm just happy we could talk about my career, that I have a career to talk about. You never know, right? Take nothing for granted.
I guess that's true. But and certainly, just looking at your Google Scholar, you have tons of papers and you're doing really well and so many different things. And maybe we'll have to have you back to talk about other things too.
I would love to do that. This is so much fun, Peter. I would love to do that. So, we have really changed our program. Our program of research, I think if there's anything that has been characteristic of our program is that there's nothing that's characteristic necessarily. That in part is a reflection of my own kind of scientific curiosity and kind of being pulled in different directions, as well as where our collaborative research has taken us. That has been, if I can kind of encourage young investigators or even senior investigators to pursue more collaboration, that's what I would do, because it takes you into areas of science where you learn and you have an opportunity to educate as well.
Yeah.
Because of this, a lot of our resources, much of our resources are now focused on this Dunedin study. And because the participants of this study are now middle-aged, they're in midlife, the focus of the research has been what can we understand about midlife that can inform their aging,
yes,
their transition into older life. And so we are now focusing on measures related to structural brain integrity, as they may allow us to have a window into later risk for dementia.
Yeah.
One of the things that I've learned about, which has been really fun and exciting, is that much of the failures, much of the disappointing results of clinical trials targeting Alzheimer's and related dementias may have to do with the fact that they've started too late in life. That when you target these interventions, whether they're physiological, dietary, pharmacological, when you target them to 70 and 80-year-olds,
it's
probably irreversible that it's too late.
Right.
It's too late. So the major shift that's happening now, a major shift, is that these interventions are being targeted to midlife.
Yeah.
But the very substantial challenge that represents is what is going to be the outcome.
Yes.
The outcome can't be Alzheimer's diagnosis, because we would have to wait 20, 30 years to know what happened. So There is a desperate need for surrogate biomarkers in midlife. And that's where we're really focusing. Can midlife measures of brain structure and function through MRI serve as useful surrogate biomarkers for intervention trials, for prevention trials? A few years, maybe we'll have more data, and I would love to talk with you about that.
That's so cool, right? I mean, you're looking at white matter integrity, but who knows? Yes. Maybe even MRI. I'm always hopeful that MRI can see a little bit more sensitively, global brain inflammation or things like that. That sort of are like you say, are sort of like indicative of your projection of how things are going.
One of the measures that we're hoping to adopt when we go back down to scan our study members, we scanned them when they were 45, and we hope to be down there again when they're 52. Is NODI or NODI? I'm not sure how it's said. This neurite orientation, dispersion, and density imaging.
Okay.
So it's as I understand it. Again, I worry that I'm going to misrepresent it. You can look at kind of a much more subtle variation in the structural integrity of neurites and kind of their orientation, the dispersion index. Yeah. What we think, or what we're hopeful for, is that while we've been able to identify measurable differences in white matter hyperintensities, which is, they accumulate with aging, they're higher in those who develop dementia, they're kind of a clinical marker. We hope that there are even more subtle measures that can be identified in midlife that can be used as biomarkers to understand risk for later dementia, and perhaps be used as outcomes in trials.
Oh, that's cool. That's very cool.
And again, we hope that functional measures can have an important role there too, because understanding the functional architecture of the aging brain is going to be really critical in better advancing the treatment and prevention of these disorders, which are only going to grow as the population continues to age.
Yeah, and I think you're on. Definitely, there's all kinds of potential indicators, and I think what you're saying makes sense. Is that it starts in your a big factor is lifestyle. I mean, certainly genetics plays a role, but a big factor is lifestyle, and that starts to accumulate after the age of 30, even, and you just start being a certain way. Then let's
not talk about it too much. It's going to make you depressed the rest of the day. No, I mean, there's some investigators who argue that your risk for Alzheimer's and related dementias begins at conception. So they're genetic factors, they're early environmental factors, they're certainly midlife factors, cardiovascular fitness. You're a runner, you know about the importance of fitness, lifestyle, diet. There's so many factors. We're hopeful that we can make some small contribution in terms of what we can learn about an individual's risk through measuring their brain.
Yes.
So far, there are some promising results with structure, and we hope that there'll be, equally, if not more promising results with function once we can move fMRI into an area where we can reliably ascertain individual differences.
Yeah, cool. Yeah, great discussion and a lot of really interesting information. And
yeah, well, I'm grateful for the opportunity to talk with you about this, Peter, and just chat. It's been a long time, and it's always fun to catch up.
Yeah, definitely. It's always fun to catch up. And like I said, I really mean it when it'd be great to have you back to talk about the other aspects of your work.
Let's do it. Now it's so easy with Zoom, with our new work life. It's a lot easier than it used to be.
Yeah, no, definitely. All right. Well, thank you again. And
my pleasure.
I really appreciate your time. All right.
It's my pleasure, Peter. It was a lot of fun. Thank you.
All right. Thank you.